lecture. We will discuss the introduction regarding the replication and the key steps of the replication and then the mode of replication, and in the last we will discuss the lytic cycle and lysogenic cycles of the replication. We can define the replication. The viruses, they are obligate intracellular parasites, which means that they cannot replicate or express their genes without the help of a living cell. Means they are not able to replicate without the host. For replication of virus, host is necessary. And viruses are host specific. Means different viruses choose for their replication for different hosts. So it is not necessary every virus can infect every cell. No. Viruses, they are host specific. Hosts may be bacteria, may be plant, may be an animal. And please mind it, viruses multiply only in the living cells, not in the dead cells. Viruses never reproduce by division, but they are replicated by the process in which all components of the viruses are produced separately and are assembled into intact virions. A single virus particle that is called as virion lacks needed components that cells have to reproduce. That's why when a virus infects a cell, it marshals the cell's ribosomes, enzymes, and much of the cellular machinery to replicate. Viral genome must be able to produce messenger RNA for replication. Now, these are the key steps in the viral reproduction. So, we have attachment, the entry or the penetration, uncoating, genome replication, assembly, maturation, release. So this is a sketch diagram is given in front of this. So you can see here, this is a cell membrane, this is a virus. Initially it comes and attaches to the receptors present on the cell membrane. Then after attachment, it is going to enter into the cytoplasm of the cell. Then in the cytoplasm, or in some cases of the viruses in the nucleus, the virus uncoating occurs. Means the capsid of the virus is removed and then this genome is free. Then this genome replication and transcription occurs. During the replication, it is going to multiply into a number of genomes, and during the transcription, the genome forms the different types of proteins, and then when these number of genome particles are formed, and along with these proteins, which are regulated proteins and restricted proteins, they combine and they become assembly. After the assembly, they become mature, and then they become mature, then they come out of from the cell, which is called as release. So we will discuss in these subheadings in detail. Starting from the attachment, the viruses are host specific and enter into the host or the target cell. This event is electrostatic, does not require any cellular or metabolic energy. Means no energy is needed for the attachment. Viruses exhibit cellular tropism. Means they have affinity. The viruses have affinity for particular tissues or the cells. Like HIV virus has the affinity or attraction for the T lymphocytes or macrophages. The rabies virus has the attraction or tropism for the muscle and the neurons. Hepatitis A, B, C, and these viruses have the attraction for the liver cells, the hepatocytes. That's why we can say the virus exhibits cellular tropism and they are host specific. Virus also have the host range, and this host range can be narrow and broad. Like the rabies virus have a wide or the broad range of the hosts, multiple hosts. They can be like the dog, the bat, the foxes. Different animals are the host for the rabies virus. Similarly, HIV virus have also the multiple hosts range. Viruses use receptors for the attachment and entry into the cell. Whenever these viruses they come, they have specific receptors on the cell membrane. Similarly, on the surface of the virus, there are also the receptors. So these both type of the specific proteins on the surface of the virus and also the specific proteins on the surface of the cell membrane, which are called the receptors. They must be present for the attachment. Cellular proteins are mostly, these cellular receptors are mostly proteins, but sometimes they can be glycoprotein, carbohydrate or lipids. You can see here, this is a cell membrane, and on the cell surface, these all are the receptors for the attachment, and this is the virus, and on the surface of the virus, see the special virus outer membrane proteins are there. So these proteins of the virus and these receptors, they attach with each other, and by this means when they interact, then this is showing the attachment. The presence of virus-specific receptor is necessary. Without these specific receptors, attachment is not possible. For example, HIV virus, they always attach to the CD4 receptors on some of the WBCs. As likewise, the viruses, rabies viruses, mostly they come and attach to the acetylcholine receptors of the neurons. So they are specific, the receptors are there for specific viruses. Cells without the appropriate receptors are not susceptible to the virus. Then comes the penetration or entry of the virus into the cell. Penetration, attachment was the energy independent, but here the penetration is energy dependent process and occurs almost instantaneously after the attachment. You can see here showing the attachment. This is the HIV virus and this is the CD4 receptor. So this is the cell membrane, host cell, and this is a so you can see specific proteins are present on the surface of the receptor and specific receptors are present on the surface of the cell membrane. So with time what happens, this protein comes and attaches to the CD4 cells initially, see here, and then what happens, these proteins also fuses with some receptors on the cell membrane, and this like this fusion occurs. Like see CD4, the protein from the this virus, and see that this protein from this cell membrane. Here the fusion occurs, attachment occurs, and this way they are going to attach on the surface of epithelium. After this attachment, what happens? They are going to penetrate within this cell membrane. So what happened? The viruses may penetrate into the host by either by the process of endocytosis, translocation, or by the process of fusion. So like you see, this is the whole process of attachment. Now after attachment, what happens? You can see here, this is a cell membrane and this is the virus. When virus comes and attaches to the cell membrane, then with the process of engulfing, now this cell membrane engulfs this virus. This is called engulfment or endocytosis. After this formation of this endosome is formed. See, this is a vesicle or endosome is formed within which the virus is present, endosome is formed. And then what happened? With time, this endosome ruptures with the rupture of the vesicle, the envelope and the capsule, they break down, and then what happens? This genome becomes free. So this is a process by endocytosis. Other way of penetration is the fusion. Now you can see here the virus comes and attaches to the fuses. This virus fuses with the cell membrane of the host cell. So see here, the spikes are present on this and the special receptors are present on the cell membrane. So these spikes and receptors, they combine each other and they form the fusion. After fusion, what happens slowly and gradually, the complex is formed by the receptor spike. So with time, what happens, this cell membrane is going to be, the opening formation is there, and the material, the genome which is present in the virus, it comes into the virus comes into the this cytoplasm, and here what happens, uncoating occurs. When the uncoating occurs, the capsid is removed and the virus genome is free. Now you can see here. So these are the two main methods by which the viruses come and penetrate into the cell cytoplasm. The third method, the third step is uncoating, which is the removal or degradation of the capsid, which is called as uncoating, thereby releasing the genome into the soil. So when the capsid is removed or degraded, the genome material, genetic material is released into the cytoplasm or nucleus of the cell. When the nucleic acid, means either the DNA or RNA, is uncoated, infectious virus particles cannot be recovered from the host. Means when uncoating occurs, now the virus become uninfectious, non-infectious. So after uncoating, you cannot recover the infectious virus particles from the cell because they have no protein coat. Now only the genome is there, and this genome is individually independent, is not infectious. That's why, and this is the start of the eclipse phase, which is lasts until new infectious virus. Means when the uncoating occurs, the eclipse phase starts, during which the virion or the particle becomes non-infectious, and this is called as the eclipse, and phase is called as eclipse phase. The virus genome is transported to the site where transcription or replication can begin. In some viruses, there is no degradation of capsid, as capsid proteins play a role in viral transcription and replication. So in some cases, there is no degradation of capsid. Then comes the viral genome replication. Viral genetic material or genome is multiplied within the host cell. Simultaneously, the viral structural proteins like capsids and other proteins are also synthesized in the cell. So type of genetic material varies from virus to virus, like it can be either the DNA or the RNA, the single stranded RNA or the double stranded RNA. So it depends, the genetic material varies cell to cell. So then comes after the genome replication, the assembly occurs. This is a stage of the replication during which all the structural components come together and basic structure of the virus particle is formed. This is called assembly. Like see here, this is the procapsid, then this is the genome. When genome comes and inside the capsid, now see, this is the now capsid, and then so it takes place at a particular site in the cell, either the cytoplasm or the nucleus. And you can see in pox viruses, assembly occurs in the cytoplasm, and the adenovirus, in case of adenovirus, it occurs in the nucleus. You can see here. So now this capsid surrounds the genome. Now this is called as assembly. After the assembly, the maturation occurs. So maturation is a stage of the life cycle at which the virus becomes infectious. Now this is the stage after maturation, means when the genome is covered by the capsid and all the assembly occurs. So now the virion becomes infectious, and this is called as maturation. Nucleic acids and capsids are assembled together. At this time, if the cells are broken, the active virus can be detected. For some viruses, maturation occurs only after release of virus particles from the cell. Next, finally, the release. The newly formed viruses are released to outside of the cell either by lysis or by budding. So, what happens? You can see in this one. Generally, non-enveloped viruses are released by cell lysis, which results in the death of the host cell. You can see here. See here, after the assembly, what happens that they are going to now release from the cell. So, either they are released by the lysis of the cell when the cell death occurs, or by the budding when there is no lysis of the cell and they are going to come out from the cell in the form of budding. Release of viruses by budding may or may not kill the cell. Generally, they are not going to kill the cell. Now, this is a good picture you can see showing the all steps. See here, initially the virus comes and it recognizes the receptors on the surface cell and then going to attach here. After the attachment, you can see here attachment, they enter into the cell. So now see here by the process of endocytosis, they are going to form the vesicle and endosomes, and then another way, if you see here, they are also showing the attachment. And after attachment, what happens? Here they are by the process of endocytosis and here by the process of fusion. So you can see that now by the process of fusion endocytosis, they penetrate. So penetration occur within the cell. So see, one method is like this and the other method is like this. This is by endocytosis, this is by process of fusion. Okay. So then after this penetration, uncoating occurs. Means now see, this coat is removed, the virus genome is released into the cytoplasm or the so what happened? This viral genome, which is most likely the DNA, now replication occurs and transcription occurs. By the process of replication, they are going to form large number of genome particles, while DNA particles, during transcription, what happens? The messenger RNA are formed and these messenger are formed. Translation occurs where the protein synthesis is formed. So different type of proteins like this, different type of proteins are formed, and in the same way, at the same time, the genome is replicated. So what happened? The capsids are formed and other proteins like these are also proteins also synthesized from this. So what happened? The assembly occurs. After the assembly, what happened? The genome goes inside the capsids. Now they are mature viruses are formed here and they lyse the cell and comes out of the cell by lysing the cell by the lysis. Other way is that these mature cells come to the surface of the cell, host cell, and here the budding occurs. In the formation of the envelope is here. You can see here the cell membrane provides the spikes or the proteins on the surface. So this is the process of budding and this is the process of lysing. It is very good explained in the next slide. I'm going to see here. See, these are the two viruses. They come and attach. So it's showing the attachment here. After attachment, what happens? See here, this is the enveloped. This is the non-enveloped virus. They enter by the end process of endocytosis. See, and when they come outside, inside they form the endosomes and the endosomes are released. Then now see, this is the unenveloped virus. So this is the enveloped virus. Now see, this is the fusion. This is the process of fusion. Then now both the viruses are within the cytoplasm. Now here uncoating occurs. You can see the coat is removed, means protein is removed. The virus genome is released. Now see that this is a viral genome DNA. Now from the DNA, messenger RNA are formed. This is called as transcription. And from the transcription, translation occurs. The proteins are formed. These proteins form different structural proteins. Now see, these are the structural different proteins are formed from through the transcription and translation. And at the same time, you see there is replication. So replication of the genome. So this is a replication. The multiplication of the genome. Large number of genome particles are formed. Now what happened then? See here the structural proteins and the genome, they combine, they assemble and form the new cells, viral cells. And then what happened? The viruses which are going to be enveloped, they come to the near, and see here the structural proteins, they are assembled here at the site of the this cell membrane. When they come out, so they will be enveloped like this. So in this way, what happened? There is no death of the cell, there is no lysis of the cell. They come out from the cell with the envelope, while these non-enveloped cells, when they come out from the cell, initially they cause a lysis of the cell, and then they come out like this. So by this way, you can understand the two methods of the replication. One is the two methods of release. One is by the lysis, in which the cells are going to die, and the other method is budding. So now the viral replication, there are two basic viral replication cycles for DNA virus, especially the lytic cycle and the lysogenic cycle. In the lytic cycle, the lytic cycle ends with the death and lysis of the cell, particularly occurs, they are showing the bacteriophages, while the lysogenic cycle, the host cells remain alive, and it is particularly seen in the lambda bacteriophage. So this is the lytic cycle. The 6 stages of the lytic cycle are the adsorption or the attachment, penetration, transcription, replication, assembly or maturation, and release. You can see here, starting from here, this is the bacterium, and these are the bacteriophage or the viruses. When they come, attaches to the here, see this is the DNA of the bacteria, and this virus when come attaches here, so what happened? They introduce, after attachment, the virus introduces its DNA or into this, and what happened here? The DNA of the virus is going to be replicate, means the transcription occurs from the DNA RNA form, and from this RNA the translation occurs, the phage induced proteins are formed. So now from these proteins, different type of structural proteins are formed here. You can see here the replication, the DNA and the synthesis of proteins, phage, coat proteins, other proteins, components and DNA are produced separately. Host DNA here is degraded. Now see, the host DNA is also degraded. So what happens when after this the assembly occurs? What happened? The different structures which are present here, this is the genome, this is the capsid, this is the other parts of this bacteriophage. Now they are going to assemble, and a large number of virus particles or phage particles are formed, and lysis occur. During lysis, all these cells, the cell is going to die, the destruction of cells occur, and large number of bacteriophages they are released, phages are released, and this is called as the bacteriophage lytic phase. You can see also this is also good. This is the bacterium, adsorption occurs. So this is the virus, attaches, injection of this is the DNA's replication of the occur, and after replication the transcription occurs, and large number of proteins are formed. Then here going to assembly, and after then maturation occurs. After maturation, what happened? Rupture or the lysis of the cell occurs, and the viruses are released, and then again the cycle starts. You can see here. Here you can see how this bacteriophage or the phage introduces the DNA into the host cell. So see, this is the DNA or the genome which is introduced into the cell. Lysogenic cycle. Lysogeny is a process by which the viral DNA becomes integrated into the host cell DNA. Replication stops and no progeny viruses are made. Later, if DNA is damaged by, for example, ultraviolet, viral DNA is excised from the host cell DNA and progeny viruses are made. The integrated viral DNA is called as prophage. So bacterial cells carrying a prophage can acquire new traits such as ability to produce exotoxins such as diphtheria toxins. Like you can see here, showing the lysogenic cycle. Now you can see here that this is the host cell, and this is the host cell, this is the viruses introducing the DNA. So this is the DNA of the virus and this is the DNA of the host cell. So what happened here, both the DNA of the virus is integrated into the DNA of the host. Now what happened, now it is incorporated into host cells. DNA is a now this is called as DNA as a prophage. It can remain dormant for days, months or even years. So what happened when the host cells replicates, both the host cell DNA and the prophage both are going to be replicated. So the new host cells, they continue to survive having the incorporated DNA, means having both the viral DNA and the host DNA. Thank you very much.